Good morning, students, or good evening, or good afternoon, or good night. I don't know when you watch and learn lectures. Today we are having Immunology lecture number 7, and the theme of our lecture is serological tests. We will talk about agglutination test, precipitation test, complement fixation test, ELISA, immunofluorescence test, radioimmunoassay, and others. So this theme was discussed firstly in microbiology a little bit. You know that in diagnosis of all infections, almost of all infections, we may use serological tests. And today I'm going to tell you and explain how we do these tests, which tests are used in bacterial, in viral infections, and in others variants. Firstly, we should say what are serological tests, about its classification and using in medical practice. Serological reactions or serological tests consists of antigens and antibodies or serum. You know that antibodies are present in serum. The specificity of immunological reactions is useful in many diagnostic tests. You remember that there is one important property of antibodies. It is specificity. It is ability of antibodies to react with only those antigens that cause their production. It is a very important property. Serology uses antibodies, usually in serum, to detect and identify antigens, or conversely uses known antigens to detect antibodies. So there will be, it may be two variants. When we have unknown antibody. What is it? It is patient serum. When we collect serum from the patient with antibodies, and we don't know what are these antibodies and which bacteria caused production of these antibodies. If we use unknown antibodies, we should use known antigen. What is known antigen? It is pure culture of known bacterial species at the laboratory. Well, for example, let's imagine that our patient has any intestinal disease, any intestinal infection, maybe typhoid fever or gastroenteritis caused by E. coli. How to identify? We should collect serum from the patient with antibodies, and we should take pure culture of Salmonella typhi, then pure culture of E. coli, mix with this antibody. If they react in, for example, in the first tube where Salmonella typhi is present, it means that in our patient Salmonella typhi cause disease. It is typhoid fever. In another variant, when we have known antibody. Known antibody, it is diagnostic commercial serum. They are present in small bottles, and it is written, for example, diagnostic serum for Salmonella typhi, diagnostic serum for Staphylococcus aureus. It means that we know what are these antibodies. These are antibodies against Staphylococcus aureus. If we use known antibody, we should use unknown antigen. Unknown antigen, it is isolated bacteria from the patient. For example, we isolate it from the patient, and we don't use bacteriological method. We are going to use just serological. We also should mix them in the tube or on the slide. If they react with each other, if they form immune complexes, it means that we may say what is species of bacteria that we isolate from patient. In this diagram you can see main principle of serological tests. It is unknown microorganism, pure culture of microorganism. This is serum containing antibodies, known antibodies, Treponema pallidum. You know, Treponema pallidum cause disease syphilis. It is the first variant. Then in the second variant we may use antibodies from the patient, unknown antibodies, and known bacteria Treponema pallidum. If they are specific to each other, they bond, and we may understand that test is positive if they bond with each other. We may use serological tests for different purposes. The first one, to identify antigen, to identify name of bacteria. What is this bacteria? In specimen, when we identify it in specimen for fast for rapid diagnosis or express diagnostics, or for identifying of antigen in pure culture of microorganism. And the another one, we may use it for serological identification to determine the species of microorganisms. It includes serotyping when we determine serological variant. I remind you that within the species of bacteria there may be serological variants. Serological variants are bacteria that are distinguished in their antigen structure. For example, structure of O antigen in E. coli. We know that in E. coli there are more than 170 serological variants. Or, for example, in Leptospira interrogans, that cause leptospirosis, there are more than 200 different serological variants. One species includes different serological variants that are distinguished in structure of lipopolysaccharide of cell wall. The another one when we may use serological identification is to reveal antibodies or immunoglobulins. We may detect the fact of their presence, it is qualitative, I say they are present or absent, and we may use quantitative test, means when we detect amount of antibodies. It is especially important in prognosis, when we detect antibodies in the first, on the third, then on the fifth and sixth days of the disease, and when we may understand if amount of antibodies increases, it means that disease develops. If amount of antibodies decreased, it means that our patient convalescent. Serological reactions may be classified into some groups. The first one is simple tests that involve just two components, antigens and antibodies or immunoglobulins. They include agglutination test or reaction of agglutination with insoluble corpuscular antigens. It means these are whole bacterial cells, and the reaction of precipitation or precipitation test with soluble antigen. They mostly use protein. The second type of serological tests are complex tests. For example, it is complement fixation reaction or complement fixation test. It is three-component test. It includes antigen, then antibody or immunoglobulin, and complement. And the another group includes reactions occurring with use of different labels. It is immunofluorescence test that use fluorescent label. I will explain. ELISA test or enzyme-linked immunosorbent assay that use enzyme label. And radioimmunoassays that use radioisotope label. The first one is agglutination test. Agglutination test. Agglutination test or agglutination reactions involve either particulate antigens, these are particles such as cells that carry antigenic molecules, or soluble antigens adhering to particles. Mostly these are particulate antigens. Bacterial cells or other particles that carry antigens on their surface. Antigen in agglutination reaction is called agglutinogen. Antibody is called agglutinin. And their complex antigen and antibody is called agglutinet. Hemagglutination reactions involve agglutination reaction using red blood cells. That's why they are called hemagglutination. Hemagglutination reactions are used in blood typing when we detect blood type AB, AB, O, I told you, in the diagnosis of certain diseases and in the identification of viruses. These antigens can be linked together by antibodies to form visible aggregates. So the positive result of agglutination test, it is formation of aggregates when antigen and antibody are bound with each other. There are some types of agglutination test. It's direct agglutination test that involve insoluble corpuscular antigen. It's slight agglutination test and tube agglutination test. Direct agglutination test is used mostly for the detection of bacteria, for their serotyping, for example. Indirect agglutination test use passive carriers of antigen to make the reaction more visible. Indirect agglutination tests are used when amount of antibodies in human serum is very, very low. It includes latex agglutination test and co-agglutination. The another type of agglutination test is hemagglutination inhibition test, and the last one is Coombs test. The first one that we discuss is direct agglutination test. Direct agglutination test. This test detects antibodies against relatively large cellular antigens such as those on red blood cells, bacteria, and fungi. So direct agglutination test is used to detect antibodies against bacteria, antibodies against red blood cells, or fungi. These reactions may be performed on slides, it's rapid test, or in tubes. In the principle of direct agglutination test, we talk about combination of a particular antigen with its antibody forms antigen-antibody complex. Look at this diagram. It is particulate antigen, bacterial cell. These are antibodies, IgM for example, that bind to antigen. And this complex is called agglutinet. We may use direct agglutination test, for example, in serotyping of Vibrio cholera or in serotyping of Salmonella. Slide agglutination test is used for rapid diagnosis. We may take a slide, place serum of the patient on the slide, And then we should add bacteria, pure culture of bacteria that we suggest could cause disease. If they form flakes, you can see these white flakes, it is considered as a positive result. In negative result, we can't see flakes. The another variant of direct agglutination test is tube agglutination test. Test serum, patient serum, is diluted in a series of tubes, doubling dilutions. You can see this. These tubes. This test is used to detect amount of antibodies, or titer of antibodies. Then, constant defined amount of antigen is added to each tube, and tubes incubated for about 20 hours in 37 degrees Celsius. Particular antigen clumps at the bottom of the test tube. Here. In this diagram, you may see positive and negative test, how to detect what is amount of antibodies, or what is antibody titer. Antibody titer, it is amount of antibodies that cause agglutination. We should prepare some dilutions of patient's serum and then add the same amount of antigen in every tube. After incubation, we should see result. In positive result, we can see bottom. These flakes that are formed in the bottom of the tube. These are positive results. These are negative. We can't see bottom here. How to detect antibody titer? We can see that this is positive result. This is negative result. So the largest dilution of patient's serum that gives positive agglutination test is considered as a amount of antibodies. So we can say then we use a formula, special formula, where we can say that antibody titer is 1 to 40. Indirect agglutination test is another one. In indirect, another name of this agglutination test is passive agglutination test. In this agglutination test, we use particles that are coated with antigens or antibodies that are normally found in the cell surface. I've said that we use passive or indirect agglutination test when amount of antibodies in patient serum is very, very small. Particle carriers that can be used for these tests are red blood cells, or polystyrene latex, or staphylococcus bacterial cells. So what are these tests? What are indirect agglutination tests? The first type of indirect agglutination test is latex agglutination. You may understand from the name of this test that we use latex as a carriers. Antibody molecules can be bound to each latex beads. Look at this diagram. These are latex, polystyrene latex, and antibodies are bound with them. When antigen is present in test specimen, it may bind to latex bead. These are antigens, thus forming visible cross-linked aggregates that we can detect. This aggregates or agglutinate is detected in the bottom of the tube. Once more, we use this test when amount of antibodies is very, very low in patient's blood. The another type of indirect agglutination is co-agglutination. This test is also used when there is a little amount of antibodies in patient's serum, and in this test we use Staphylococcus bacteria as a carriers. This test is based that killed Staphylococcus aureus has a protein on its surface which binds antibodies. Any antibodies against Staphylococcus or against other bacteria may be bound with their Fc receptor. Fc, you remember structure of immunoglobulin, yes? It is Fc. Any antibodies may be bound with protein A of Staphylococcus aureus. So Staphylococcus aureus cells are coated with antibodies and mixed with unknown antigens. In positive result, we can see agglutination. I have just said about indirect agglutination. Another type of agglutination reaction is hemagglutination inhibition test. In indirect method for detection of viruses is to search for viral antibodies in patient's serum. Certain viruses like influenza, measles, or mumps have ability to agglutinate or form clumping red blood cells. This phenomenon is called hemagglutination. So some respiratory viruses have spikes with hemagglutinine on its surface that provide clumping of human red blood cells. It is called hemagglutination. But our test, this test is called hemagglutination inhibition. Why? What is this mechanism? Let's look. The patient's serum is combined with known viruses to which red blood cells are then added. If the serum contains antibodies for that virus, the antibodies coat the virus, and when the viruses are then combined with red blood cells, no agglutination will take place. Let's look at this diagram. These are known viruses, for example influenza, and these are antibodies from patient's serum who we suspect where we suspect that patient has influenza. If they are really specific to each other, if these antibodies against influenza virus, they are bound to each other. Viruses are coated with antibodies, but we can't see it. We add red blood cells, any human red blood cells, and red blood cells are not bonded to viruses because all viruses are busy with antigens. Hemagglutination inhibition, so absence of agglutination of red blood cells means positive result. It means that these are antibodies against influenza virus. In another state, in negative result, what happens? For example, let's imagine that we suspect that our patient has influenza, but and we take influenza virus and collect serum from the patient, and we can see and then add red blood cells. And red blood cells are agglutinated. It means that antibodies and antigens and viruses are not bound with each other. and viruses, free viruses, are bound to red blood cells. It means negative result that these antibodies are not against influenza virus. Coombs test is another variant of indirect agglutination. Coombs test is used to identify rhesus negative mothers who are producing anti-rhesus antibodies. You remember we talked in lecture number 4 about such disease, about such allergy that is called cytotoxic hypersensitivity. It occurs in rhesus negative women and leads to hemolytic disease of newborn or erythroblatosis fetalis. Because mother's immune system produces antibodies against baby's red blood cells. How to detect, firstly, how to prevent hemolytic disease of newborn? We should detect these antibodies. Mother's serum containing anti-rhesus antibodies is mixed with red blood cells that have rhesus antigen on their surface. So we should collect serum from mother and mix it with rhesus positive red blood cells. Then incubate it, and during this process, during incubation, antibodies bind to red blood cells, but we can't see it, and we should add rabbit anti-human immunoglobulin. What is it? It is a serum from rabbits whose organism is immunized, is injected with such antibodies. And in rabbits' organism, its immune system produces antibodies against these antibodies, because these antibodies are considered as foreign for rabbit. That's why immune system of rabbit produces B cells of rabbits produces antibodies against these antibodies, and this serum that is produced, that is got from rabbit, agglutinate or bind to mother's immunoglobulins, and just in this case we can see agglutination or clumping. The another type of serological test is precipitation reaction or precipitation test. Precipitation test involves the reaction of soluble antigens like proteins and toxins, unlike agglutination test that I have just explained, where it uses particular antigens or cellular antigens. Antigens and antibodies rapidly form small antigen and antibody complexes. It may be done in tube, so it is called ring precipitation test, or in petri dish, in agar, it is called gel precipitation test or immunodiffusion. Antigen in precipitation reaction is called precipitinogen. Antibody is called precipitin. Complex of antigen antibody is called precipitate. The first variant here is ring precipitation test. It is very easy to understand. We should take tube, we should add serum of the patient, and then we should add serum of the patient with antitoxic antibodies, and then we should add soluble antigen, it is toxin or any protein. when they react to each other, we can see precipitate. It is a ring, white ring, visible line or white ring. Gel precipitation, or it is also called immunodiffusion. Immunodiffusion because antigens and antibodies are diffused into agar. Immunodiffusion tests are precipitation reactions carried out in agar gel medium on either a Petri plate. Wells or holes are cut into a plate of purified agar. Different known antibodies. It means that we know what are these antibodies against which bacteria or virus. Bacteria mostly. Different known antibodies are placed into two upper wells, and a mixture of unknown antigens is placed into the lower well here. These reactants diffuse into agar from the wells and form cloudy lines, cloudy white lines. They react here, antibody diffuse here, antibody diffuse here, antigen diffuse here, antigen diffuse here. When they form immune complexes, we can see cloudy lines, white lines. The lines cross each other because each antigen has reacted only with its complementary antibody. So if we, for example, add antibodies against E. coli, and these, for example, antibodies are against Salmonella typhi, let's imagine, and here are unknown antigens, are known culture of bacteria isolated from patient. If we can see white line just here, it means that the patient is infected just by E. coli. If we can see white line is here, it means that patient is infected just by E. coli. If we can see white lines in both next to the both wells, it means that patient is infected by both bacteria. Neutralization test is used to detect bacterial toxin. I've said that in precipitation test we detect soluble antigen. Soluble antigen may be protein or toxin. So for detection of toxin, we use neutralization test or neutralization test of toxin by antitoxic serum. There are two variants that may be used to detect bacterial toxin. The first one is made in vitro in the tube. It includes flocculation test. Flocculation test resembles ring precipitation test. When toxin is added in the tube, then antitoxic serum is added. If they are specific, they flocculate and form visible cloudy floccles, like clouds. And another variant of neutralization test in vitro is ELIC, gel precipitation test. I will explain what is it. We also may do neutralization test in vivo. In vivo means in lab animals, like in mice. Firstly, about ELIC gel precipitation test. We talked about it when we studied airborne infections, Corynebacteria diphtheria, for example. ELIC gel precipitation test is used to detect toxin of Corynebacteria diphtheria. How is it made? Filter paper strip, please look at this diagram. Filter paper strip containing diphtheria antitoxin. Antitoxin, it is a serum with antitoxic antibodies, serum with antibodies that are produced against toxin. Filter paper strip containing diphtheria antitoxin is placed in the center of agar plate. Bacterial strains of Corynebacterium diphtheria from patient, then this is from patient, then known positive bacterial strain, for example here, and non-negative toxigenic bacterial strains are streaked on the agar surface in a line across the plate, like here, and like here. So this is unknown bacterial strain isolated from the patient. This is known positive, because we should compare them, and this is known negative. Non-toxigenic means that don't produce toxin. After some time, antitoxin or antitoxic serum diffuse away from the strip, where as a toxin produced by bacteria, by toxin produced strains, diffuse away from growth, from culture here. At the zone of equivalescence, a precipitin line, this one, is formed. If this line is formed, means this strain is Corynebacterium diphtheria that produces toxin. How is it visible in Petri dish? Look at this diagram, at this photo. This is filter paper strip containing antitoxic serum. This is positive known Corynebacterium diphtheria strain that produces toxin. This is negative Corynebacterium strain that doesn't produce toxin. And then these are strains of bacterial Corynebacterium diphtheria that are isolated from the patient. Here we, for example, can see that they study two strains. And what can we see? We can see precipitin lines here, because it is positive result, and here that means positive result. That means that this strain produces toxin. This one doesn't produce toxin. We can't see white lines here. Another variant of neutralization test is made in vivo. In vivo means in living animals, in living organism in animals. Actually, we may use mice in most cases for it. Firstly, in every experiment, there must be a control group, or like here I have explained it, control. Positive control, negative control, because we need to compare with something. Our results of our experiment must be compared with something positive or negative. In the neutralization test in vivo, we also use control group of animals when we inject specimen with bacteria producing toxin into animal, and in this case mice died because of bacterial toxin presence. In second group of mice, we should inject mixture of specimen with antibodies against toxin A. It is an example of botulism. You remember this disease, botulism. There are some strains of Clostridium botulinum that produce toxins. These strains are called A, B, E. These are the most dangerous. So we inject specimen in mixture with toxin A. We inject it into mice, and mice are alive. It means that in its organism, toxin was neutralized by antitoxic serum. This toxin from bacteria was neutralized by antibodies. That's why mice are alive. In the third group, we inject mixture of specimen with antibodies against toxin B. what happens here? We can see that mice died. Why? Because toxin was not neutralized by antibodies, by antitoxic serum. It means that in specimens, it means that in specimen it is not toxin B, that bacteria don't produce toxin B. And in the fourth group, we inject specimen together with antibodies against toxin E. Mice also died. It means that in this case it is the same, toxin was not neutralized by antibodies. They are not specific to each other. It means that these antibodies are not against this toxin. So by this method we can detect which toxin is produced. The another method is a complement fixation test. It is a complex method. Complement fixation test can be used to detect very small amounts of antibodies. The complement fixation test is performed in two parts. The first part is called a test system that uses a patient's serum or antibodies and antigen and complement derived from guinea pigs. I hope you remember what is complement. It is plasma proteins produced in liver that are present in human organism in non-active form. It is activated just in the presence of immune complexes, complex of antigen and antibody. The second part of complement fixation test is called indicator system. It requires sheep red blood cells and antibodies that recognizes the sheep red blood cells. So in the second part, in the first part, pay your attention, there are antibodies, antigen. In the second part also there are antibodies and antigens. These are red blood cells. Firstly, I want to explain it is the text, and then I will show you the diagram, the scheme of this method, because some students understand just from the text, some students understand just from scheme, from the picture. Its mechanism. Firstly, in the first stage, antigen, then patient serum and complement are added to a test tube and incubated. And then the another indicator system is added. It's sheep red blood cells and antibodies that lies sheep red blood cells if complement is available. What happens in the tube? If the patient's serum contains antibodies to the antigen, these antibodies bind antigen, they form immune complex and activate complement. And you remember that complement is activated C6, C7, C8, C9 that cause lysis. It is a membrane attack complex that causes lysis of this complex. But we can't see it. We can't see this mechanism, this process in the tube. This process is not visible by a naked eye. To see it, we should add sheep red blood cells and antibodies to these cells. They also form antigen-antibody complex. If antigen-antibody complex is formed, complement must be activated. But what can we see? Is complement present? No, complement was used in previous stage to destroy antigen-antibody complex, of serum, antibodies and added antigen. That's why complement has been used up in the first stage, and we can't see lysis of the sheep red blood cell. There is no lysis. No lysis of sheep red blood cells occurs if primarily human patient serum contained antibodies against antigen. In another variant, if antibodies from the patient's serum are not present in the serum, no reaction occurs and the complement is free. Antigen and antibodies are not bound with each other, complement is not activated and it is present in the tube. Then we add sheep red blood cells and antibodies, they form immune complex, and it activates free complement that destroy sheep red blood cells. So we can see lysis of sheep red blood cells if primarily antibodies were absent in patient's serum. Look at this diagram, look at this scheme. Once more, it is the tube. We add patient's serum with or without antibodies, we don't know. Antibodies are present or absent. Then we add antigen of, well, for example, it is bacteria. Noun culture of bacteria, noun antigen. And then we add complement. We incubate during 90 minutes at 37 degrees Celsius. If antigen binds to antibodies, if antibodies are present, they activate complement, and then we add sheep red blood cells with antibodies to these sheep red blood cells, and they form immune complexes and must be destroyed by complement, but complement is absent. So we can see that red blood cells are not destroyed, are not lysed. No lysis means that antibodies were present primarily in the tube. Antibodies were present in patient's serum. If antibodies were absent, what happens? Antigen is free. It's patient serum without antibodies. Complement is free, and when we add sheep red blood cells with antibodies, complement is activated and destroy sheep red blood cells, and we can see lysis. Complement fixation test is a complex test that is used in diagnosis in serological reactions. Another one is another group of tests that are tests that use different labels or antibody binding. The first one, or labeling methods, the first one is immunofluorescence test. Firstly, detection of antigen and antibody binding can be enhanced visually by attaching a label to the antigen or antibody. So any label may be attached, may be joined with antigen or antibodies. What are these labels? It may be fluorescence dye that is used in immunofluorescence test. It may be enzyme like peroxidase that is used in ELISA, and it may be radioisotope that is used in radioimmunoassay. The first one is immunofluorescence test. Immunofluorescence test is used for identification of microorganisms in clinical specimens and detecting of the presence of a specific antibody in serum. So we can identify as a microorganism as antibodies in serum. Fluorescence dye, such as fluorescein or rhodamine. Fluorescein gives green color. Rhodamine gives red color. These dyes may be attached to non-specific antibodies, like here, which are then used to detect the presence of antibody in serum or microorganism in a sample. Fluorescence antibody technique may be direct or indirect. In the direct method, the fluorescence dye is linked to a known antibody. For direct testing, antigen is fixed to the slide. Antibody to which fluorescence has been attached is added. When viewed with a fluorescence microscope, antibodies appear yellow-green. Let's look at this diagram. It is an example of direct immunofluorescence test. This is antigen, for example, streptococci from throat that cause sore throat disease. And these are antibodies that are labeled with fluorescence dye. If streptococci are present in specimen, in throat discharge, in throat swab, they are bound with antibodies, and then we can see in fluorescence microscope their appearance. You remember the streptococcus are cockle-shaped bacteria. They occur in chains, so we can see that they occur in chains. So you can see green-yellow appearance of streptococcus. So it is used for detection of bacteria in specimen, in clinical specimen. Indirect fluorescence antibody test is used when the fluorescence dye is linked to an antibody recognizing human antibodies in a patient's serum. I understand it is difficult to memorize and to understand. Once more, in the previous one, indirect fluorescence dye is labeled with antibodies. In indirect, fluorescence dye is linked to and antibodies that recognize human antibodies in patient's serum. This method is used to detect antibodies. If direct method is used to detect antigen, bacteria in clinical specimen, indirect is used to detect antibodies in serum. Known microorganism, it is antigen, is fixed to the slide. For example, this one, known Treponema pallidum, is fixed to the slide. A sample of test serum antibodies is added, and the mixture on the slide is incubated. We take serum from the patient who we suspect has syphilis. We mix them on the slide and incubate. If antibodies are present in this serum, they will bind, look here, they will bind to the antigens of the organism. But we can't see this result. We can't see it by naked eyes. We should add something that allows to see this result. What should we add? We should add anti-human gamma globulin. Anti-human gamma globulin consists of antibodies that may be bind to human antibodies. And this anti-human gamma globulin must be labeled with fluorescein or rhodamine. It is also added to the slide. These antibodies bind to human antibodies, and then the slide is observed using the fluorescence microscope, and you can see the presence of antibodies by fluorescence. Sure, we can see shape of bacteria, because antibodies cover all the bacterial surface. It means that patient's serum contains antibodies against known bacteria that we used firstly. The another labeling method is enzyme-linked immunosorbent assay, or ELISA. In this method, there is also two variants. It is direct ELISA that detect antigens, and indirect ELISA that detect antibodies. Mechanism of ELISA, direct and indirect, also depends what we are going to detect. If we are going to detect antigens, we should use serum, noun serum that is labeled with enzyme. If we are going to detect antibodies in patient serum, we should use anti-human, anti-globulin serum that is linked with enzyme. You'll understand what I mean. You will understand, I will explain. Antigen absorbs to the plastic surface and the excess is washed away. The test serum is added then on a plate and incubated. If specific antibodies are present, they bind to the antigen. To detect the antigen-antibody reaction, their complex, the ELISA uses anti-human gamma globulin chemically coupled with an enzyme label, peroxidase. Once more, when antibodies and antigens are bind to each other on a plastic plate, we can't see their complex. We should add anti-human gamma globulin that consists antibodies against human antibodies that is labeled with enzyme peroxidase. They binds to humans antibodies. Then we should add specific substrate. It is chromogen. Actually, chromogen is a colorless substrate that produces a colored end product when acted on by enzyme peroxidase. Chromogen is fermented, is destroyed by enzyme, so we can see changing of color. This color change may be measured, and it's directly related to the amount of antibody bound. Let's look at this diagram. I hope you will understand. Firstly, for ELISA, we should use plastic plate. This is, or micro titer plate. It is called plastic plate. We should add. Firstly, we should add antigens, unknown antigens. Known antigens in this plate. Then we should add serum from the patient, in every variant, in every hole. We can't see antibodies are bound with antigens or are not bound with antigens. We should add anti-human gamma globulin serum that is labeled with peroxidase enzyme. And we also can't see anti-human gamma globulin are bound with human antibodies binded that bond firstly with antigen or not bond. We should add chromogen. In every variant, in every hole, we should add chromogen. In those cases where anti-human gamma globulin is bound to patient serum, we can see changing of color from yellow to blue. So in this case, we can see positive result. It means that in this variant, patient serum contains antibodies against, for example, against HIV, like here. ELISA tests are useful in studying of infection diseases, and modification of ELISA tests are available commercially to test for group A streptoculture. Today, streptococcal ELISA tests are used for rapid diagnosis of sore throat in the children. We may buy it even in pharmacy. They resemble pregnancy tests. They are also like paper strips. We can just touch by this strip the throat discharge of the child, and after some time we can see changing of color of in positive cases if streptococcus are present in the throat of the child. It is also used to detect antibiotics, to detect cytokines in human blood. Pregnancy tests are also based on ELISA. Presence of drugs in urine we also may detect by using of ELISA. The last labeled method is called radioimmunoassay. It is extremely sensitive serological procedure used to measure the concentration of very small antigens. This technique is used for diagnosis of hepatitis antigens, reproductive hormones, insulin, and certain drugs, even for trillionth of a gram of a substance. Antigen is bound to a plastic device, such as a plastic plate with wells. The patient's serum is added to the device. Look here in diagram, added. The antibody may or may not be present in the solution. An antibody that reacts with a human antibody is added. It is anti-globulin antibodies or human antiglobulin serum. But these antiglobulin antibodies must be labeled by with radioactive label. What it may be? Different radioactive isotope. Look here, it is anti-human antiglobulin serum, and this is isotope. They are labeled with isotope, and if antibodies are bound, human antibodies primarily bound to antigens, they are bound with anti-human antibodies. What is the result? What is positive result? We can see radioactivity. It is detected in the wells. The technical concludes that the solution containing the suspected antibodies. In negative result, radioactivity is not detected. And the last test that is used in diagnosis, it resembles labeled methods. It is called Western blotting. Or another name of this method is immunoblotting. Immunoblotting is used to identify specific proteins in a mixture. Proteins. Firstly, I'm going to explain, then I will show you the scheme diagram. Antigens of microorganisms, proteins or glycoproteins, for example, in HIV. Immunoblotting is widespread for diagnosis of HIV. Antigens of microorganisms are separated by electrophoresis in polyacrylamide gel. So antigens are separated into fractions by their molecular weight. Then antigens are placed on nitrocellulose membrane. Membrane is coated into strips. The patient's serum is placed on the strips and incubated. The enzyme-linked anti-human antiglobulins are added. It resembles ELISA. The substrate are added, and we can see the number of colored fractions or spots that correspond to the localization zone of the antigen-antibody complex. Let's look on this scheme to understand it. So, firstly, once more, immunoblotting is used to detect proteins or glycoproteins, antigens of some virus, HIV, for example. Antigens, firstly, are separated into fractions. Viral antigens are separated into fractions by electrophoresis, and then they are placed on the surface of nitrocellulose membrane. Look here, this is nitrocellulose membrane, and there are some strips, and we place a certain antigen in one strip. A antigen, another antigen. A, B, C, D means different antigens. The first, the another antigen. For example, let's imagine that we isolate 7 antigens, different 7 antigens from HIV virus. So we place them on the surface of nitrocellulose membrane and then add patient's serum that must contain antibodies. Add on this to this nitrocellulose membrane. Then we should patient's serum may contain or may not contain antibodies. Then we should add enzyme-linked anti-human antiglobulin serum that is added, and that should be bound with human antibodies, with these antibodies. We can see it in this picture, in this stage. And we don't know these anti-human antiglobulins are bond or are not bond with patients' immunoglobulins, with patients' antibodies in serum. We should add substrate. We should add substrate, and then after some time we can see changing of color because of destruction of this substrate by enzyme. So, and we may say which antibodies were present here, because in some strips we can see, we can see changing of color. It means that in patient's serum contains antibodies that were placed here, for example, and here. Actually, in every strip, antigens must be placed. These are just examples. So we can say which antibodies were present firstly. This is a result of immunoblotting. For example, these are antigens. HIV virus contains a lot of different antigens. They are mostly proteins. So finally we can see that there is a positive result in this antigen, It means that firstly in patient's serum there were antibodies against this antigen and against this antigen. Now that's all. Thanks for your attention. Learn this lecture for next immunology class. Next week, I remind you, next week we will have my